Welcome to this video where we will be introducing some of the basics of proteins. So pause this as needed to read through these objectives. It's primarily going to be looking at the structure and functions of proteins. So you should be able to do all these things by the end of the video. So the first thing that we're going to do is go over how proteins are made. So proteins are made up of building blocks known as amino acids. They get their name from their structure. So an amino acid has an amino group, that's what's in this blue box here, as well as a carboxylic acid group that's in this red box. You will also find a central carbon that is common to all the amino acids and a hydrogen that comes off from them. Now where the amino acids differ is in this R group here, which can have a variety of different groups that give the different amino acids their different properties, which ultimately are going to give proteins their different properties as well. So if we look at some of these, notice down here we have glycine, one of the most basic ones. It just has another hydrogen coming off from this. But we do have ones like this one over here that has a chain of carbons and then an amino group over here. So this is one of the basic amino acids, so it has properties of a base. This is one of the acidic amino acids. You can see a carboxylic group again, so it's going to have properties of an acid. We'll have some that have rings on them that give them special properties, and other ones that have other groups such as this OH group over here. Now these amino acids are going to be combined in a linear fashion, so how that's going to work, if we take two of these amino acids, so we're taking this one over here with the ring and the one with the OH group, and you put them together in a ribosome where they can react, or where the cell puts proteins together, you're going to have, you'll notice these HOH groups. So this, if you would take that out, that would be a water, so it's called a dehydration reaction because it removes these, essentially removes the water from the molecule combining these two. So now you have what's known as a peptide bond. So this is going to link the two amino acids together. And then this can happen again in the ribosome, and again, and again, and again, 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 and you eventually have a long polypeptide chain that can be put together to make a protein. So we're going to look at the primary, secondary, tertiary, and quaternary structure of proteins. So each of these little blocks represents a different amino acid, and the primary structure is simply the order that these amino acids go in together. So this one might be an alanine, then you have another alanine, then a proline, then a cysteine, etc. So primary structure is just the order of the amino acids. Now the secondary structure is how these amino acids fold in three-dimensional space. So it's the individual amino acids interacting with each other that give them their secondary structure. Now there's two main structures that you will find with that are associated as secondary structures. One is alpha helices, and the other is beta pleated sheets. So this is just common ways that they fold together. Is found in many different proteins. Now the secondary structures, then there can be several of them within a protein. So you can see there's an alpha helix here. Here's a really not well defined region of the protein, and then another alpha helix, and then you come over here and you have a beta pleated sheet, and then you come over here, have another beta pleated sheet, and then ending with an alpha helices. So these secondary structures then can fold together and give a three-dimensional shape in space. So if we were to do that, this alpha helices is moving over here, this beta pleated sheet is moving over, and then you might have more folding such that you have a three-dimensional structure. So this is how the different secondary structures are interacting in three-dimensional space to give you really a folded protein structure. For some proteins, this is it. This is how the protein is folded, and you're done. Other proteins are going to be made up of several of these subunits. So you would have one polypeptide chain here, and another polypeptide chain over here, and they could come together and give you a three-dimensional protein by interacting. So quaternary structure is when there's more than one polypeptide chain interacting to give you the three-dimensional structure. So this entire folding process gives the protein its specific function. So how the protein is folded functions on how other proteins recognize it, how it's interacting in the body, how it can actually function. So there are ways of making the protein unfold, and that's what denaturation is, when you have the protein unfold. So if you increase the temperature too much, this can mess up with the interactions that are holding the protein together and cause it to unfold, which is what's being shown down here. There's changing in the acidity of the environment, so your stomach acid can denature some proteins as well. Salt content, organic solvents, other things can also do that. But temperature and pH are usually the two big ones that people generally think of. And so when the protein unfolds, it loses its function and can no longer function properly to give the protein its structure. Sometimes when you remove the denaturation agent, so if you turn the temperature back to normal or you return the pH to normal, the protein might refold to its state, but this is somewhat uncommon. This is why once you fry an egg, it goes from being, the egg whites go from being clear to being a white shape because the proteins are denaturing. And then you let the egg cool, it actually doesn't renature until it's folded shape. So how it is originally folded is sometimes important for the protein to function. So what are some functions of proteins? A better question would be, what don't proteins do? Because they're involved in pretty much everything that your body does. The majority of what you are looking at when you're looking at an individual are their proteins. So their hair, the skin cells have a lot of protein that are making them together. So a lot of different things are going on. So they can act as enzymes. So an enzyme is a protein catalyst, essentially. So it's something that helps aid the metabolic reactions that go on in our body. They can act as transport proteins, either as something that's going to move things across the cell membrane or something that's going to move substances within your body, such as the hemoglobin in red blood cells. They play a big part in immunity as antibodies. They're very important for structure. Notice that your hair is made primarily up of keratin and the proteins that really give it structure. Support, so there's the exoskeletons that support our cells, as well as the proteins that are associated with your skeleton and many others that are going to give it its function as well. It can be as an identification marker, so there can be proteins that are on the surface that say, hey, I'm this kind of protein. It can also act in communication, so receptors can respond to hormones such that your body can then react. All of this is related to homeostasis, just meaning balance then. And then finally, probably the most commonly thought of function of protein is movement, muscle, getting along, but it's also cellular movements of things within the cell. So this is just a kind of a brief summary of the functions of proteins. They really are a diverse category, and it's really the structure of the protein that's going to give them all of their diverse functions.